This is about controlling microbial growth through things like autoclave and disinfectants. So if you say sterilization is removal and destruction of all microbial life, and then the caveat to that is it neutralizes virus, right? You don't necessarily, I guess destroying it doesn't mean you kill it. The spores that bacteria make are very resistant to heat, so you need to make, conditions in the autoclave, able to destroy bacterial spores, right? So you wouldn't need an autoclave necessarily. If you boil water, for instance, that will kill most bacteria, but it won't kill their spores. So it's safe probably when they say to boil your water for, if they've had a problem with the water line or something, in case there's bacteria in it, but it doesn't sterilize. So autoclave does. It wouldn't remove, or it wouldn't prevent you from getting a problem with a prion, which is not a big problem anyway in the United States. But it would probably remove virus. Disinfect. This is what you use, say, with disinfectants. Right, if we use this, it probably destroys, 99%, but it doesn't eliminate everything. And they normally tell you how well I noticed this one doesn't tell you, but a good one would tell you exactly what bacteria or virus it kills and at what percent. It's usually above 99%. So asepsis is a body surface instead of a inanimate object. And then sanitization just means that you're using a soap or washing a surface, which is kind of what this does. Although you're supposed to spray it on and not wipe it off. So soaps are kind of like sanitization by removing things. If you refrigerate, you say it has a bacteriostatic effect, and that just means it's slowing it down from growing, right? It's not dividing as quickly. That's the idea behind milk, right? You keep it in the fridge. If you leaved it out for a couple hours, then the shelf life would be reduced, right? It just means there's more bacteria growing. When you pasteurize, which we'll talk about, milk, you can't really autoclave it, because if you autoclave milk, it would destroy the milk, right? It would curdle. Now, if something is bactericidal, then it can kill bacteria. It has to be sporocidal, though, to kill spores. Now, it doesn't mean that if you use this that it wouldn't kill bacteria, but the word itself means it's specific to virus, fungi. But I would think most disinfectants would be all four. High pressure, that's what an autoclave gives you. And then this is the big table, right? Physical, chemical, mechanical. We'll you can filter a fluid instead of autoclaving it to remove virus and bacteria because they can't get through the filter. So this is an example. Don't worry about all these different times, but moist heat means that you're applying steam. Dry heat, no steam. So you can see by having the steam in the autoclave that it takes a lot less longer for it to sterilize. So that's why when you use an autoclave, right, it's heat, steam, and pressure, all three used. And you need a chamber to pressurize. So you put something in here, even liquids, and you might have to keep them in there for about 20 minutes. And then when they're done, right, it's heat stabile. Stabile? stable. If it's heat labile, that's the opposite. Heat labile means by adding heat to it, it destroys. Like that's what would happen to milk. If you put milk in an autoclave, it would curdle. Basically, the protein in the milk curdle. That's what curdling is. So if it's heat labile, then we'll use a filter. I'll talk about that. So when you think about a disinfectant, one of the ways it can work is through inactivating proteins. So on the top, it just shows a folded protein, and then these are, I'll say, completely denatured. I'm not sure why they changed the color to yellow, but it's this guy now that's unfolded. It can't do whatever the protein is intended to do. So the two main categories would be enzymes and I guess that, and then structural proteins maybe that sort of keep the virus or the bacteria, the structure of them. But by changing the pH or adding heat to proteins, usually cold temperatures aren't a big problem, but they can be. But when you change the shape, then the protein can't do what it does. Even heavy metals, I added oxidation to this because when you oxidize proteins by adding electrons to them, it has a similar effect. It changes the shape. And proteins can only do what they do by having the correct shape that they need. That's how they get things done. So we left off here, and I was saying that metals are very antimicrobial. And then there's ionizing radiation, and then UV radiation is considered non-ionizing. Ionizing radiation, examples of that are things like X-rays and gamma rays, and they can be used to sterilize food. It actually sterilizes. And these type of radiation can penetrate packages like even cartons of milk, but you would consider that ionizing. UV radiation, like you can have what are called UV hoods, or maybe even a UV hood, where you have a light that's emitting UV light, but it can cause DNA damage, but it doesn't sterilize. If you turn it on while you're not using the hood or the room to reduce the amount of microbes that could be a problem. And then, as I said before about the heat labile means that the heat will hurt you. If you can't autoclave a solution, then we talked about counting bacteria using filters. So if you run this fluid through the filter, as long as this was sterilized prior, then everything that falls into it through the filter would be considered sterilized. Bactericidal chemicals, we're going to get into another thing, bactericidal or disinfectants. We say they kill. Now, they don't sterilize even if they're cidal. They kill bacteria, and there's always, say, 99.98% effective. That means most of the bacteria are dead. But you can't assume that they all died. And if you look at a disinfectant label, it may list the different things like E. coli, HIV, and they may not be the same. But they'll normally list on the disinfectant how effective they are. If they don't have this on the disinfectant, it probably isn't. The phenolic, it's plural. right? These would be different chemicals that would be considered a phenolic-like disinfectant. Right, and they kill. Here's some more examples of disinfectants: chlorine, iodine. Iodine's a pretty good one. Alcohol, hydrogen peroxide is very good. Now they're listing, what's weird is like they say, some of them are bactericidal, virucidal, some are fungicidal, and then they put on here sporicidal for hydrogen peroxide. Now it's bactericidal too. I don't know why on this they don't say that. I don't know why. Sporicidal is effective against spores. More than likely, if something is sporicidal, then it's probably going to kill the bacteria as well. But you can't assume that. It is called Mueller-Hinton plate. And what's done is you cover the entire surface of the plate, you coat it with bacteria. Before the bacteria grow, you put on paper discs that contain, in the Mueller-Hinton, those discs contain antibiotics. Now, in this experiment, they put the quaternary amine. They put, right, probably peroxide, I think is what the chlorine did. Hexachlorophene, 4 different disinfectants. So each disc contained one of those 4 disinfectants. And you would assume that the disinfectant would probably go into the media to some extent. Hard to know, but you assume that too. Then you incubate the plate and let the bacteria grow. Right, and then you would assume after that, this area here, the bacteria are growing, and then the darker area around the disc, there is no growth of bacteria, right? You call that a zone of inhibition in this context. And you could assume, right, Staph aureus was put on this plate, they put E. coli on this plate, and they put Pseudomonas aeruginosa on the third plate. And then you could say, how effective are these 4 disinfectants against these 3 bacteria in these circumstances? And you could say probably all 4 were effective against Staph aureus just based on the zone of inhibition. But E. coli, right, hexachlorophene didn't work. Right. So the other 3 worked, but really not, you know, a little bit less effective than this. But you probably couldn't make any conclusion to that. But Pseudomonas was resistant, and it has been that way for a long time, is more resistant to the disinfectants than the other 2, or other bacteria might be. They're just known for their ability to be resistant. So if you work in a hospital, healthcare, I guess that's not associated infections or nosocomial infections. And Pseudomonas has always had a reputation for causing many of them, because when you disinfect the rooms in the hospital, it's more likely to remain behind than other bacteria. And here, 3% hydrogen peroxide works pretty good against these 3 organisms, and fairly quickly. When you use a disinfectant and you spray it on, or before you use it, it'll tell you, if you read the label, it'll say, this must be left on for 30 seconds on the surface to be this effective. That's the other thing that tells you that the disinfectant is probably pretty good if it gives you a time point. So really the best way to use that, and the way when I use it and spray the tables, is I spray it on and I don't wipe. It stays on the table longer, right? If it's there for 2 minutes, that's pretty long. Now, if you use ethanol, it'd take 10 minutes to kill Staph aureus. But you need to leave your hand in alcohol. You have to dump it in there for 10 minutes, really. So it's not that effective, really. Don't worry about all those times. I'm not going to test you on that. Now, quaternary amine, this is what that means, right? Amine means you have a nitrogen in the middle, and then quat means 4. So there's 4 things attached. And then germicidal soap means that it contains a disinfectant versus not soap. I kind of like to show you this to say that if you're using soaps without disinfectant, it's just removing. But that's probably, you know, as good as, you know, having the disinfectant in it helps, but, you know, you can't say, well, now my hands are free of germs either. You still have to. Then this is a list of common products like Tilex or Lysol, and then what's contained in them. So this is the disinfectant, ethanol, triloscan is in this. Hypochlorite is like a bleach. Right, this is the category, right? These are all phenolic, right? Triclosan is a phenolic compound. But I'm not going to have you, you know, I won't ask any specific questions on this from this table. Okay, so that's section 10.